For a scientist, the most exciting thing is to see a problem and find a solution.
Creativity, scientific rigor, and discretion. That's what characterizes Dirk Görlich. This scientist comes from Halle in Germany, and since 2005 he has been conducting research at the Max Planck Institute for Multidisciplinary Sciences in Göttingen. His specialist area is cell transport, and in particular the mechanics of protein exchange within cells between the nucleus and the cytoplasm.
If one chooses a topic for a PhD or for a postdoc, it has to be an open question. And protein transport into the cell nucleus was at that time the most unexplored topic.
Cell nuclei hold our genetic data, and it is essential for them to be able to export and import proteins to and from the cell cytoplasm. This exchange takes place via nuclear pores, which are present on the envelope of the nucleus.
Our starting point was to measure the process, and we could calculate how many shuttles does a nuclear pore complex carry per second, and the numbers were amazing. At this point, we could invert the question, how comes that normal proteins do not get into the nucleus? And this was when we started thinking of there might be a barrier.
This barrier is today known as the FG phase. It is a physical structure which Dirk Görlich's scientific teams have been able to describe and reproduce in the lab.
The material that forms this barrier is actually similar to something everybody of us knows in real life. It's like a gummy bear. We found how to get such material in our hands. All the information for making this material is written in a rather simple sequence. You just have to bring these molecules, these FG repeat domains, to a sufficiently high concentration, and they form a material that just behaves like a nuclear pore complex.
Basically, in the central channel of the nuclear pore, you have a meshwork, and this mesh or this sieve doesn't allow everything to go through it, so it's selective.
This is something we now understand. Are the two compartments kept separate, and how does a cell achieve that just the needed proteins get into the nucleus, and how does a cell achieve that, say, messenger RNAs can get selectively exported from the cell nucleus?
Dirk Görlich's teams are now focusing on understanding how this selection takes place and how certain molecular structures manage to breach this natural protection system.
How the material can pass through this FG phase is a very important process in terms of the cellular transport. For a virus to infect human cells, for example, so some particle of the virus has to pass through the FG phase. If we know how this process happened, we may be able to block this viral transfection.
Among the viruses which have been examined so far, HIV is an example of one which manages to disguise its genetic material and covertly introduce it into the cell nucleus.
The strategy of HIV is not to expose its genome to the cytoplasmic antiviral sensors. It keeps the genome all the time in there until it has docked to the nuclear pore complex and transported the contents into the nucleus. So the antiviral sensors have no chance to see the dangerous payload.
Thanks to biochemistry, understanding such subtle mechanisms is the first stage of extending the horizons of our knowledge. By uncovering these cell transport processes, Dirk Görlich has shown the way forward and brought into being a new field of science.
Is not only important for itself, it's a phenomenon. There is a spread of his work, so to speak, into medicine. I also think that he was the person not only who had the idea, who developed the model, but he really showed it to the point. And I consider him the father of this field.
it really helps to understand how in biology and in life and in these cells specifically, they've developed these really tiny microsystems where a lot of biological activity and reactions are contained to a specific space within the cell that is not surrounded by a membrane or any physical barrier. More and more groups are now saying that actually their processes and the things that they are also working on also takes place and is localized with a similar mechanism. I think that this is kind of where the knowledge is then translated throughout the field.
Many people are working on this. It's an exciting field to see phase separation being important for many, many cellular processes. And the FG phase at nuclear pores was just the first of its kind.